This is All Things Cancer, the podcast of the National Foundation for Cancer Research, hosted by NFCR's president and CEO, Dr. Sujuan Ba. Dr. Ba is a globally recognized cancer research advocate and visionary leader. All Things Cancer episodes will explore the hindsight, insights, and foresight of cancer. From research discoveries to patient care advances, and how NFCR and our collaborators are working to accelerate the process of making cures possible. In this episode, Dr. Sujuan Ba sits down with Dr. Drew Pardoll, one of the world's leading voices in cancer immunology. From his groundbreaking discovery of key immune pathways to his role in shaping biotech startups that translate science into real treatments, Dr. Pardoll's career embodies the power of innovation and collaboration. Together, they explore the promise of engineering proteins, cells, metabolism, and even microbes as the frontier of cancer research, while reflecting on the vital role of young scientists and the importance of bold, unconventional thinking. Let's dive in.
Dr. Pardoll, your career covers such a wide range of disciplines. How do you transform into different fields and make such a profound impact for cancer patients?
I never expected to be an immunologist. I trained with a molecular biologist and geneticist, Bert Vogelstein, and also Don Coffey, who's a cell biologist. And I had sort of one of those transformational moments when I was an oncology fellow on the bone marrow transplant service. We used to take care of the pediatric transplants, and the adult transplants were all on the same service. And back then, it was typical for the patients to be in the hospital for months at a time. Not so anymore. So you really got to know the families and the patients very well. One of the toxicities of bone marrow transplantation was graft versus host disease. It's when the T cells, the immune system of the bone marrow graft, is transplanted into the patient. They recognize the patient's tissues and organs as foreign and can attack the patient. Could be very terrible, but we were able to tell the patients and their families if they survived their graft versus host disease, their cancer would never come back, because that immune system was also creating a graft versus tumor effect.
There must be a hopeful message for cancer patients and their families.
Absolutely. So you could see the power of the immune system, and right at that time, to keep my science brain going, I would get copies of various of the journals. And I opened up a journal one night. It was about 1 o'clock in the morning, taking care of patients. And there were the articles from two former St. Georgie winners, Tack Mack and Mark Davis, on the first cloning of the genes for the T cell receptor. And that was a watershed moment in immunology, where it went from a cellular and a phenomenologic field and discipline really to a molecular discipline, that you could apply molecular biology. And so I said to myself, this is going to be the way that we can ultimately fine-tune the immune system, use its power as a weapon against the tumor, but more specifically so we don't have all of the damage against the patient. And
so that was your aha moment.
That was absolutely my aha moment. I remember it distinctly. I decided I wanted to train in basic immunology before going into cancer immunology. As an oncology fellow, there was no T-cell immunology at Hopkins at the time. And so our cancer center director allowed me to do my research years down at the NIH, learning basic immunology. And I always am wearing my basic immunology hat, and we've stumbled onto some very interesting cells and pathways, including the second ligand for PD-1, now called PD-L2. And that pathway is obviously very important in cancer. And the same time, my wife, Suzanne Topalian, had just come to Hopkins, so she and a young faculty member, Julie Bramer, put together the clinical trial. We were all working together with a small biotech company called MedRx. And the beauty of cancer immunology is that almost everything that you learn in the immune system is relevant to the immune system's interaction with cancer and therefore relevant to cancer immunotherapy. Mm-hmm. And so you're simultaneously wearing your immunologist hat and your immunotherapist hat as you're in the laboratory, and the ability to see the discoveries translate into patients. It's over many, many years, but it's really worth it.
You have founded or co-founded 7, 8 biotech companies, and one of them actually NFCR provided the seed investment, the Monty Bio initially named, and now it's called Collapse Therapeutics.
Yes.
And so tell me more about those exciting biotech startups that you have founded and co-founded, and how they transform your research in a way.
So, when I started on the faculty, it wasn't something that I was thinking about, but what I learned is that there are certain things that companies do better. An idea, a discovery, proof of concept gets to a certain place in the lab, and then it goes into a mode where the investment is larger. You have to worry about good manufacturing practice, all of the sorts of things that really don't work as well in the laboratory. And it was early times where biotechnology startups were occurring, and it really became a great opportunity to sort of take that intermediate step between the laboratory and ultimately when something is going to be used in hundreds, thousands, tens of thousands of patients, that generally goes into pharma. But the biotech startup is a really good opportunity to do that. In the case of originally Manatee, now Clasp, this has been particularly special because I founded this together with my PhD advisor, Bert, and one of my
NFCR worked with Bert for many years.
NFCR worked with many years. Ken Kinsler, and also one of my former postdoctoral fellows, who's now an associate professor, Kelly Smith. And the first employee, when it was originally Manatee, was an incredibly talented graduate student that Kelly and I co-mentored. Her name is Justina Kaushi. Her PhD thesis was a first author paper in Nature. And so Clasp has 4 scientific generations.
That's fantastic.
I think that's
a great story.
I defy any other biotech to be able to come up with literally 4 scientific, and we're all very much involved. We are extremely engaged with the company. To my mind, I think it represents the paragon of what a biotech startup should be in terms of the input from both sides. We, the academics, are seasoned enough that we appreciate the things that they really do well. They've been employing some phenomenal protein engineering approaches, including some of the approaches that the Nobel Prize was given out for protein engineering, some of the AI-based protein folding and structure design programs to take the sort of initial, call it lead compound that was made in the laboratory that we published together in Science in 2021 and turned it into a drug now. And it's the first IND is going to be filed. This is using the immune system to target the most common oncogenic mutation in P53.
And that's a hard one to target.
So it's very hard to target with drugs.
Yes.
Very, very hard. Remains to be seen if ammunition. But this is the immune system. This is T cells recognizing the peptides that contain the mutation in P53 that are presented on HLA from the tumor. So again, it's sort of the perfect integration of cancer genetics and immunology to the point where they're really not distinguishable subfields anymore. It's really all one field.
The science are all converging together now.
Yeah, I mean, and half of Bert's group now is doing immunotherapy engineering. So I turned him into an immunotherapist.
And you've been collaborating with Bert and Ken for decades. Tell me about your trick of getting all those wonderful people to collaborate with you.
Well, it certainly helps that they're all a lot smarter than I am. So I can hitch my caboose to a fast-moving train. It's a very familial kind set of relationships. When I was a graduate student of Bert's, I had an experiment going all day, and I didn't have time to go to the cafeteria. I would just go into his desk and steal his lunch. So actually,
but I heard that he only has very simple lunch: sandwich and tomatoes and cheese.
When you're in the middle of an experiment and you've got 2 minutes to spare, that's all you want. You don't want anything. So that's absolutely true. You don't want anything fancy. The problem is, at the time he was kosher, so he couldn't. When I stole his lunch, he couldn't go to the cafeteria to get lunch. So one day he actually put a tarantula in his lunchbox just to sort of teach me a lesson. That's actually true.
That's a wonderful story.
But the reality is that there's a lot of, we are very much family. There's a lot of humor that goes into it, and we just have a lot of fun working together, and also bringing very different perspectives. So it's the ability to have different perspectives, to have relationships such that you can have really vigorous debates and disagreement.
And that's why they are so productive, very proliferate in terms of making discoveries.
And a lot of that is brainstorming. The whole idea to use anti-PD-1 in this genetic subset of patients that are mismatch repair deficient, or MSI, which was in part the geneticists' discoveries, Bert's discoveries and his colleagues, Nick Papadopoulos and Ken, that these tumors have 50 times the number of mutations that a normal tumor has that doesn't have that mutation. And so that makes it much more viewable by the immune system.
Yeah.
And so all you have to do is just empower the immune system by blocking this one break on the immune system, PD-1. That came out of a pure brainstorming. It started in part because we had one colon cancer patient that had a complete response to anti-PD-1, and the next 15 or 20 didn't. And so we were puzzling over that. Bert, Luis Diaz had this idea of, that melanoma is responding well to anti-PD-1 because it has a lot of mutations, because it's UV induced. And I said, well, yeah, but what about this colon cancer? Colon cancers don't have a lot of mutations. And it was a brainstorming session, January of 2012. And the idea just popped out of that brainstorming session. We went back, tested that patient's tumor. Lo and behold, it was that 5% that are mismatch repair deficient. Did a trial.
Given you work with so many young scientists, do you have any concern about how their research will be funded in the future?
The tremendous constrictions in NIH funding now, particularly for young people in the field, make it difficult to do some of this brainstorming, risk-taking. This is why NFCR funding, because you guys, aren't so rigid about, do you have this, that, and the other preliminary data? Do you have an interesting idea? And, the folks that review that, the NFCR grants, are looking at it from the standpoint of, is this a really interesting idea? Are these scientists going to be able to pull it off? If it doesn't work, it doesn't work.
Our funding philosophy from day one is different from the traditional government funding. We believe that we need to give the scientists the funding so they can really explore. And when they are able to explore, then the breakthroughs can come out. And that has been our philosophy.
This, I think, is a national concern about the long-term health of academia, particularly the young people, because it's the young people who are coming in, who have the energy, have great ideas. Working with graduate students and fellows is really the most fun part of the job, because they're just looking forward to all the opportunities that the new technologies and advances are revealing, particularly to be able to make discoveries in patients. Mice are still a very important part of an immunologist's toolbox, but when I look at the number of experiments at our publications 25 years ago versus now, so much more is able to be done in the patient, to be able to study patients that are undergoing immunotherapies. Some respond, some don't respond, trying to understand why. However, doing that work is expensive, and we really worry about the young scientists, and so I think support for young scientists is important. Those of us who are older and grizzled, I think it's our mandate to be supporting young scientists. Certainly at Hopkins, that's always been the culture.
I know Johns Hopkins has a very strong mentorship program, and we, NFCR, have been really facilitating the collaborations. And now we are giving the grants based on the team they put together. Ideally, we like to have established researchers like you, and then team up with young scientists. That would be, like you said, generational inspiration to grow the scientists and keep them in the science field.
Very much so. Very much so. I think we have that obligation. Certainly NFCR's focus on support for young scientists, folks like, Dr. Zarif, Dr. Yates, is fantastic. The early days of Manatee,
yes,
which was, that was the seed funding that you guys were involved in. It was 6 people.
Wow.
It was 6 people. And, one of whom was our former graduate student, who's absolutely amazing. Absolutely amazing. I mean, she did experiments for her PhD thesis that I would look at those experiments and say, I could not have pulled that off. So it was, a small thing. We didn't have a website. It was, they were in a lab in the incubator, the biotech incubator building called Fast Forward, that's on the Hopkins Medical School campus, which was actually very convenient because our cores allow startups, folks from startups that come out of our Hopkins laboratories, and fee-for-service basis to use the cores. Yes, and this was a real innovation from Christy Weisskeel, who heads the JHTV, Johns Hopkins Tech Ventures, actually co-chaired when she first came. Our university president asked me to co-chair a committee to create a white paper on how Hopkins, which always had great academics, could really up their entrepreneurial game. And first thing I said to Christy was, to me, a white paper is something you work hard on and nobody reads it. So she says, what would I have to do to convince you that this white paper that this committee generates actually had meaning? And I said, show me blueprints for an incubator. And I'll never the day she called me up. She said, Drew, can you get over here? I have something to show you. And it was blueprints, and that was the Fast Forward building. So Manatee started up there when it was very small. It ultimately, particularly with the Series A, outgrew the space and then moved out to Rockville, which is exactly what a company does when it's successful and it gets more funding and grows. But this is now an iterative process. Actually, Bert, myself, and some incredibly smart junior faculty probably about to do another one.
Oh, wow. And you have to let us know.
Yeah, and I mean, if you trace it back, it started with that small group. And so this is really super fast.
So that would be your number 9? If this new one coming out?
I've sort of lost count.
I counted from the public search.
It's somewhere.
It's like an 8 and counting and going.
It's somewhere in there. This one is definitely special. The other thing, and I know you are involved with Blackbird.
Yes.
I've gotten to know Matt, actually, who I first met through NFCR.
I took him to meet you. He heard about you, but he never met you. I said, come along. I'm visiting Drew and Suzanne. Come and join me.
Yes. So they're actually supporting an early stage project. These are, again, high-risk projects. So it was so exciting that Blackbird was, put some support into this. And again, this is a project that you wouldn't yet write an NIH grant on. It's now going to get there. But the bottom line is that this couldn't wait for, the process that takes on average a year and a half. Usually it's a second submission.
And also they might think it's too risky, too early. So,
yes.
So I'm glad Blackbird is supporting that project.
Yeah, and they're focused on junior faculty early stage projects in Baltimore, Maryland area, which is great. I've been a Ravens fan for back to when they were the Colts.
Wow, that's a long time.
It broke our hearts when they left town. But these are the sorts of investments in young people in early stage ideas that really are what drives the future.
So Maryland, Baltimore, the ecosystem is still not there yet compared to other cities, other life science hub.
So Blackbird comes from, Edgar Allan Poe, as in the Raven, grew up in Baltimore.
Interesting. I didn't catch
that's
that.
Why the team is called the Ravens,
Ravens.
and that's what Blackbird refers to.
My vision was that we need a core group of people, catalysts, that we can work together. So you can have multiple nucleus, right? Then you can form an ecosystem and nurture all the young scientists and the young entrepreneurs and nurture the biotech hub. Maryland wanted to do this for so many years. We need to work together. We hope to do more, to work together. I'm all about bringing people together, bringing organization together, bring teams to work with a common goal.
I figured that out. The number of connections that find their way back to you is breathtaking. It just is, and so NFCR is the catalyst is the perfect term.
Yes.
It's the perfect term. You all have catalyzed a lot, and these are going to turn into a startup. I think it's very promising. 5 years from now, this thing is going to be taking off. It's going to be going into the clinic. There are going to be antibodies against this target, and you're going to look back and say, that was a no-brainer 5 years ago. I came to Hopkins in 1973. I was accepted at the ripe old age of 15 as an undergrad. And so I've lived the major part of my life in Baltimore. Did all my training at Hopkins. I've basically had one job my whole life. They change the title every once in a while. But I like to say that doing science is like being on the Orient Express. You're on the same train, but the scenery is always changing.
I love that.
So that's what being at Hopkins for me was. The scenery is always changing. And the field is always growing and changing, even if you're at one institution. So I'm very much a Baltimorean at heart. And so it's great to see some of the commitment and investment at the catalytic stage.
What do you think is on the horizon, the breakthroughs to solve today's unmet needs for cancer patients? And I also wanted to hear what are the most proud accomplishments that you have made, pushed out of those biotech arena?
So as far as the future is concerned, I would encapsulate it in the word engineering. We, the field, are able to dissect apart tumors down to all of the 25 different cell types in the tumor, take each of those, subset them based on their transcriptional program. So we are learning a lot about targets, about the pathways, the biology that is associated with resistance versus susceptibility to, in my world, immunotherapies. That's now translating into engineering problems. So protein engineering, bispecifics, trispecifics, taking pieces of proteins that are part of nature in our genome, but putting them together in new ways to endow new functions. And again, I think very apropos that the Nobel Prizes for protein engineering,
Yes.
because those platforms make, something that would have taken 3 years, you can literally do this in a month or two. So that is now huge. Cellular engineering, been around for a while, took a while to sort of get off the ground, but massive opportunities there. Still a lot of innovation, I think, over the next few years to make it easier and feasible, less expensive, more scalable. And the two other things I'll throw out in the engineering category are metabolic engineering. Mm-hmm. We're now understanding so much more about how metabolism not only is central to the tumor, but also to the immune system, which is why my former Hopkins classmate and residency mate, he is now in our Bloomberg Kimmel Institute for Immunotherapy. Yeah, so that's one of the major programs in our institute is immune metabolism. So this is a cancer immunotherapy institute, and it's immune metabolism. That can now be engineered. You can engineer that. You can make prodrugs that of inhibitors of certain metabolic pathways. Mm-hmm. This was actually one of the spin-outs that came out of our institute, not from my lab. Originally, Jonathan Powell went into a company called Drayson, and that's actually now being tested in a very interesting, important, infrequent cancer called fibrolamellar cancer, but it afflicts teenagers. Mm-hmm. It's a liver cancer, and 95% lethality. There may be an answer there in metabolic engineering. And the final thing is microbial engineering. So there's been a lot on how the microbiome and components of the microbiome can affect the immune system systemically and obviously response to immunotherapies. It's not necessarily going to be particular species of microbes in the microbiome. It's going to be particular metabolism of the bugs that is actually affecting the immune system. And so you can actually engineer the bugs. I mean, bacteria are easier to engineer than...
Cheaper, probably.
A lot cheaper.
Yes.
And Hopkins is actually starting a trial with an engineered bacteria that makes muramyl dipeptides, which can activate myeloid cells to enhance immune responses in the gut. There's evidence that because the immune system is a wanderer, T cell that's in your gut today, 2 weeks from now is going to be in your tumor. Mm-hmm. And so those engineering components, protein engineering, cellular engineering, metabolic engineering, and microbiome engineering, I see that as really the explosive future for the next 5 years.
And you mentioned the trials, right? So I've seen a lot of trials reported at the meetings, failed trials. No durable results, and no substantial survival. And I sit there and I felt so sad because every failed trial scatters all the hopes of those people in the trials. I mean, they are not subjects, they are all human beings. So I felt that we as a community, medical community, we need to do a better job designing clinical trials. Better use of the resources, the time, and also don't waste any patients. So do you see any new signs that emerging that really help to design better trials so we can get results quicker and use less patients in there, give patients more hope?
So I'll answer that question this way. You're absolutely right. There is the occasional home run. PD-1 was a home run that it was obviously incredibly exciting to be a part of. But there, I think the MSI story is, I think, the key exemplar of how clinical trials can be made better.
The biomarker.
The biomarker. If not for the insight that brought the science together with that one colon cancer patient. To this day, nobody would be treating any colon cancer patients. And that 5% of colon cancer patients, half of which are cured when it's stage 4. When you do it earlier, this was Luis Diaz's trial of neoadjuvant, 14 out of 14 patients were cured
And
when
that's
selected
miracle.
for that
Yeah.
biomarker.
Exactly.
So you hear companies, you hear pharma, they always say biomarker, biomarker, biomarker. But you then actually look at the trials and you say, relative to the money that was thrown into this 800 patient, $190 million, $250 million phase 3 trial, plus the phase 2, how much was really invested in early biomarker analysis? And was it being done correctly? Was there thought put into it? When we do an experiment in the laboratory with mice, we put a lot of thought into it. And a lot of time goes into sitting, and saying, what's the question we're asking? What are the controls? What are we going to analyze in these mice that we treat with this particular agent? Yes, we measure, how many, tumor size, but there's so much more to it. And that's with inbred mice. There is not enough careful thoughtfulness put into biomarker analysis at the early stages, in my opinion, particularly when an area gets hot, a particular target gets hot. There's a target now for regulatory T cells, these called CCR8, which can be expressed at higher levels on subsets of Treg cells. 7 pharmaceutical companies are doing clinical trials with depleting anti-CCR8 antibodies that I know of. And
right now, ADC, hundreds of them in the pipeline for clinical trials.
Right, because you had a few successes, which are exciting, and that falls into the protein engineering piece of this. And there's no question, the platforms to make these antibody drug conjugates are improving and improving. They're really great creative platforms on the technology side. But okay, here's the target you're going after. What's its expression on normal cells? What are you looking for? How do you select patients? It's not like it's zero, but in my opinion, if you look at the proportion of the overall investment, particularly when you consider how many of these multi-gazillion dollar investments do go down in flames, with a lot of patients who going on a trial, they're putting their lives
online
in
our hands.
And I just think we could do better.
I absolutely agree with you. Designs smarter trial. Biomarker is an important tool to guide us to design better trials. And that's why NFCR was the early founders for the GBM Agile, that platform. We actually provided initial leadership and the funding when other organizations don't believe in that because it was too difficult to bring everybody together to think systematically, push the boundary and keep asking those harder questions. But now look at it. We have 7 arms using this system for GBM and going to be applied to ovarian cancer and pancreatic cancer. They are all designed to use biomarker, statistical analysis, real time to guide the trials. We need more and more systems like that. We need to do better science and better biomarker research.
Yeah, arguably I'm biased being in the academic world, but I think that if companies engaged academic scientists who we just think more about this kind of thing, if we were engaged a little bit more, I think the companies would potentially do better. I think certainly, particularly in pharma, the paradigm is you design the trial internally and then you go to various institutions and get oncologists to put patients on the trial. The input from the academic sector as trials go further along is, I think, undervalued. I think particularly in pharmaceutical companies, which have thrown a lot of good money after bad with failed trials.
And that's actually one of the purposes of our global summit and our ceremony every year. Our meeting is designed different from most of the meetings. People come here not just doing the results reporting, because a lot of those results reporting you can get online. I want to put people together as a think tank, really challenge each other, stimulate each other's thought, and provide more provocative suggestions and ask hard questions, so that a lot of people who come together at this meeting will be able to take insights and new ideas going back to whatever they are doing. So that's my goal. But I would love to hear from your perspective. You have been coming to this meeting for quite a few years, and what do you feel?
This meeting is very different, which is what makes it so much fun. And I think that, first of all, it's a mix of outstanding basic scientists, translational investigators, clinical investigators, and you bring in people from the private sector, certainly a lot of biotech, but also pharma, engaged in a format where people really are not just getting up and, give their talk, get back in the taxi, go to the airport. I mean, it's really configured that it's what I told one of my colleagues to describe this meeting, an immersive experience. And that's what I think makes it a lot of fun. There's just a lot more brainstorming and going on. And, also your spirit is very uplifting. Both realizing that we still have so much more to be done that needs to be done, while at the same time celebrating our successes and that balance. But to me, it's bringing all of these different sectors, people from different perspectives together, again, in a format where it's not just one talk after the next, but panels, discussions. It's an immersive experience. And that's a real minority. There aren't enough meetings like that.
Even every, sit-down dinner we have, the table, the group, it's curated with right group, with the people who wanted to collaborate with who. So I get a lot of requests.
I've noticed the non-randomness.
Yes,
of
nothing is random. Yeah,
it's a lot of fun. I mean, these meetings, there's so much that comes out of just the interactions at the meeting. I mean, I can think of a couple just from the past few meetings. I mean, this meeting is on the do not miss list.
Wow.
You schedule it early, you put it on your calendar.
That's fantastic to hear. That it's very gratifying, and all my team and all my partners, when they all work very hard. Come here, you are immersed with all types of people, and you are challenged to think new things.
Totally. Keeps my brain exercised.
Well,
and I mean, you can see there's an energy in the
It's
meeting.
a different energy, right?
Yeah.
This meeting, each year putting meetings together like this is one of the most gratifying, satisfying, fulfilling moments.
Well, it's great that you feel that way as well, because you and your staff put in an amazing amount of work in putting this meeting together.
I'm so glad to hear.
Thank you for listening to this episode of All Things Cancer. Be sure to subscribe so you never miss future conversations. To learn more about how the National Foundation for Cancer Research is working with the global research community to make cures possible, visit nfcr.org. You can also reach us directly at 800-321-CURE.